Okay, today we're going to be talking about the nervous system and nervous tissue. So, what is the nervous system? The nervous system is a really fast communication system in our body. And in fact, when we say fast, we mean like microsecond, not like milli, but micro. And so that is like, milliseconds. A millisecond is 1/1000th of a second. All right, so a microsecond is 1/1000th of a millisecond. That's super fast. And that allows our body to get information from the external environment, but also integrate that with information regarding things in the internal environment of our body, like our blood sugar concentration or sodium concentration. The integration of sensory information is super duper important. In other words, what you're looking at and what you're hearing needs to be integrated so that you can appropriately respond to what's happening in your environment. The nervous system helps us coordinate our voluntary and involuntary motor activities. Motor always means that you are moving, okay? And then it controls and regulates other tissue systems so that we can maintain homeostasis, which is what we talk about a lot in physiology. So what you're looking at here in this picture is a human brain. Now, obviously, this human brain is no longer alive because it's outside of the body. It's a beautiful structure. This brain, or in this picture of this brain, you can see two discrete regions, something called white matter, if I can get my pen to work, there we go, and gray matter. This is really important. The white matter are collections of axons, or axon bundles. When we say bundles, what it is an axon is an extension from a nerve cell, which I'll just draw out kind of like that. And you have multiple nerve cells sending axons to other parts of the brain. These together are going to be the white matter. And you'll see later on in some drawings what's happening with those axons or nerve bundles or white matter in the brain. Now, white matter in the brain is very similar to a nerve out in the body. Just remember that right now. Gray matter are the cell bodies. So this is the gray matter, and the white matter are the axon bundles, or this part right here, okay, the nerves. So gray matter, cells, white matter, axon bundles, or nerves. In the brain, always call it white matter, though. So there are two distinct sections of the nervous system: the central nervous system, CNS, which consists of the brain and the spinal cord, and the peripheral nervous system, the PNS, which are cranial nerves and spinal nerves. These are those long axons extending out to other parts of the body to take information out for voluntary movement and to bring information in, which is sensory information from the environment around us. When we get to sensory systems in the next module, we're going to talk a lot about spinal nerves and a little bit maybe about cranial nerves. Now, there are two ways to break down the nervous system organization. There's the structural organization, which we just talked about, and then there's the functional organization. So the functional organization of the peripheral nervous system specifically are the afferent systems, which is sensory, and this is what we're going to call info in. Afferent is always info coming in. Efferent is info or information going out. Okay, so afferent is information coming into the body. Efferent is information, or I guess afferent is into the brain. Efferent is info out of the brain. The efferent systems are broken down into somatic, soma, right? Remember that, meaning cell body, which controls skeletal muscles. So that is motor information. Autonomic or automatic, auto, right, controls smooth and cardiac muscle tissue and glands. Now, autonomic is interesting because this is all the automatic stuff that you do not have control over. This is all involuntary. Remember the different kinds of muscle, which are smooth and cardiac? Those are regulated by autonomic nerve responses. Autonomic is further broken down into sympathetic and parasympathetic. Sympathetic is fight or flight, and parasympathetic is rest and digest. So sympathetic and parasympathetic are antagonistic actions within the autonomic nervous system. So when the hungry bear is coming at you, or the big hungry cougar, that's a sympathetic fight or flight reaction that your nervous system is going to induce to get you out of that stressful situation that might be, terminal for you. Sympathetic nerve activation can also be activated through exercise, heavy exercise, but also through illness, like if you catch the flu. Sympathetic activation actually helps coordinate activities in the immune response. We're not going to go into that in really great detail, but there are some immune cells that react to fight or flight. Parasympathetic, rest and digest, is hopefully what you're going through right now. You're learning, you're not very stressed out. Sympathetic activation, rest and digest, is where your body is basically going through and doing regular activities to maintain homeostasis. And so here is one way to look at the nervous system. Central nervous system is the brain and the spinal cord, super important for information processing. Oops, I'm sorry. All right, so we can call the brain and the spinal cord an integration center. Motor commands go through the efferent division. Remember, info out, efferent is out. And that's going to activate things within the peripheral nervous system, somatic, autonomic. Somatic is always skeletal muscle, soma for muscle cell. Okay, soma, remember soma is cell. And then autonomic, which is parasympathetic or sympathetic, which is going to be regulating smooth muscle, cardiac muscle, and glands. Sympathetic, fight or flight, F or F. Parasympathetic, rest and digest, R and D. Okay, rest and digest, not research and development. All of this stuff's going on over here. Now, for the afferent information coming in, we have receptors, visceral sensory receptors that monitor respiration, cardiovascular response, blood pressure, digestive processes, urinary, reproductive processes. That goes straight into the afferent division, which is going to send information to regulatory centers and integration centers in the brain. But then we have special senses and somatic sensory receptors. We're going to go into these much, much more in sensory systems, but these all bring information into the brain. So here's just a quick sketch from your book. Central nervous system consists of brain and the spinal cord, and the peripheral nervous system are basically everything outside of the spinal cord. Information coming in, information not necessarily going out, but you can have loops out in the peripheral nervous system. We'll talk about that later. Another picture showing us the brain, processing power and so forth, the spinal cord. Exiting the spinal cord are the spinal ganglia. Ganglia are collections of nerve cells that collect information from sensory structures. So when you feel, let's say, pressure on your skin, peripheral nervous system ganglia will respond to that pressure. It helps us relay information and integrate information. We don't need to worry about that too much quite yet. The nerves in our body are those long axons, and some of the nerves are going out, which are efferent, right? Some nerves are coming in, which is afferent, right? And so these long nerves, remember, are white. Nerves look white, and we'll talk about why later, but also nerves are the same thing as white matter inside of the brain. The digestive tract here is an example of what we're going to call an enteric nervous system, an ENS. Now, an enteric nervous system is basically a group of cells that regulate an organ outside of the brain. So, for example, our digestive system has autonomic functions that operate without your brain. In other words, what's happening is we have digestion, absorption, and contraction of the smooth muscle that occurs independently of your brain regulating it. So yes, the brain will help regulate the pace at which our smooth muscle will contract. However, one of the things to be aware of is when you remove that, when you remove regulation from the brain to smooth muscle, one of the things that happens is that it kind of starts contracting peristaltically on its own at a different pace. If you put more food into your digestive tract, it starts contracting and undergoing peristalsis at a different pace. And the reason that happens is because it's regulating itself independently of what the nervous system is telling it. So there are some parts of the body that do some of that. In animals, a great example of that is activation of ovulation by penetration in cats, dogs, and rabbits. So some animals, upon penetration of the male into the female, that actually activates ovulation. They're opportunistic ovulators. That is not regulated by the brain. It's not actually even regulated the same way as it is in humans by the endocrine system. It's a physical stimulation that causes ovulation. And in that case, that is an enteric response. So let's talk a little bit about what neurons look like and a little bit about also some of the vocabulary nomenclature. Nucleus. The word nucleus has means the center of something, literally, but we use the word nucleus in a lot of different ways in science. So nucleus of an atom is the center of an atom that contains protons and neutrons. You guys remember that from chemistry. Everything else are the electrons and space around it. In a cell, the nucleus is an organelle, and it's an organelle that contains chromatin, which is DNA and its associated proteins. There are also the beginning pieces of ribosomes and RNA in there. In the brain, the nucleus is a group of cell bodies, a group of neurons that live in the same space and kind of do the same job. They work together. So over here in this brain scan, you are looking at two individual nuclei. Those nuclei send axons or fiber tracts to another part of the brain. And so each one of these regions, the cells within each one of these regions cooperate to do a given job. Now, one of the things I like about this picture is you can see the white matter really clearly. Look at all of that white matter. The stuff above it is the gray matter right here, okay? That's in the cerebral cortex. One of the things that's fascinating, at least to me, is look at how much cable is running through the brain. A lot of our brain space, our brain volume, is taken up with white matter. And those are all the connections of the cells talking to one another throughout the brain. This region right here is a pathway that connects the two hemispheres of the brain. There are a couple of them, but this is the thickest one. It's called it's the cerebral commissure or the corpus callosum. I'm sorry, I need more coffee. Corpus callosum connects the right and the half hemispheres of the brain, and it's basically allowing the two sides of our body to coordinate. So now let's talk about the organization of neural tissue. There are two types of cells within the brain and the spinal cord. There are glia and there are neurons. We're going to start with glia, which some people call neuroglia. I mean, I don't know why you say neuroglia, because the only part of the body that has glia is the nervous system, so just call it glia, but whatever. Glial cells are supporting cells, and we have a billion of them in our brain. Glial cells are much smaller than neurons. They are able to undergo mitosis and cellular division, so they can proliferate. And they do a few different jobs. They act as, originally they were thought to act as glue, holding neurons together. They do separate neurons from one another so that they have adequate spacing. They give the brain a framework, a place for all the neurons to sit comfortably, and they do act as phagocytes. They can clean up, let's say if somebody gets a concussion and there's a very small bleed in the brain, the glia can clean up red blood cells that bleed out into the brain if there's just a very, very small break in the blood-brain barrier. They can also absorb compounds like free radicals that neurons spit out endlessly. Neurons are super hungry, very, very metabolically active cells. These guys clean up the area around neurons. And there are different kinds of neurons that you find in the central nervous system and the peripheral nervous system. So let's start with looking at the central nervous system, brain and spinal cord. We have ependymal cells. These are cells that line the lateral ventricles and the central canal of the spinal cord. These little feet on the end aid in circulating cerebral spinal fluid. You'll see that in a little bit. We have microglia, little dinky guys. They get rid of debris, dead cells, any pathogens that might get into the brain via phagocytosis. There are some cases where there are pathogens that can get in, there's a really gross amoeba that can get into your brain. The microglia can clean those amoeba up if they interact with them. Astrocytes, they're called astrocytes, astro for star, because they look kind of like stars. These are the cells that make the blood-brain barrier. They give structural support to the brain. They do regulate the amount of sodium and potassium and calcium outside of neurons in the central nervous system. I am not going to talk about the action potential and how it works, but you need sodium, potassium, and calcium for the activation of neuron action potentials and for the release of neurotransmitters at the synapse. So the astrocytes help regulate those ion concentrations. They also help reabsorb and recycle certain neurotransmitters. Now, these guys don't necessarily recycle serotonin, but they do recycle, I think, GABA. And then we have oligodendrocytes. Oligodendrocytes make myelin in the brain and in the spinal cord. The cousin of the oligodendrocyte is the Schwann cell. A single Schwann cell wraps around an axon, okay? So this would be the axon. This is the Schwann cell, and here's the Schwann cell's nucleus. Each Schwann cell makes a single piece of myelin that insulates peripheral axons, okay? So this is a part of the peripheral nervous system. So both Schwann cells and oligodendrocytes do the same thing. It's just one Schwann cell wraps one axon, and a single axon can have multiple Schwann cells on it. On the other hand, oligodendrocytes, you can have one oligodendrocyte interacting with 2 axons or 3 axons. So this also helps aid build a structural framework for the brain, holding neurons in place and axons in place. So you're going to find lots of oligodendrocytes in that white matter. So we talked about the Schwann cells already in the peripheral nervous system. The last type of cell we're going to talk about are satellite cells. You don't always find these in high densities throughout the peripheral nervous system. You do typically find them in the ganglia associated with the spinal cord. So they surround neuronal bodies. They do regulate oxygen and carbon dioxide levels, and they can also regulate neurotransmitter levels. They can absorb neurotransmitters like acetylcholine or norepinephrine, and they can influence the behavior of neurons inside the ganglia based on their regulation of neurotransmitter levels. So let's see what these guys look like. So we have microglia, the little tiny guys, the small cells that live inside of the brain mostly. Motile or mobile. They crawl around in your brain. Isn't that kind of creepy, but kind of fun? And they're phagocytic. Ependymal cells, I did want to show you those ependymal cells. This is the central canal in a section of spinal cord. So here's a drawing of a section of spinal cord. And in the center of the spinal cord, so this is the actual spinal cord. Around this is the vertebra. Okay, so this is living tissue. This is bone on the outside of it. All right, so what we often look at the vertebra, when we're looking at the bone of the vertebra, you're looking at the vertebral body. Remember, the vertebral body has a small, in fact, I even drew this wrong. Let's get rid of this. I'm going to draw it so that it's upside down. Remember, there's that foramen in the vertebral body, and then you have the big piece of bone down here. Here's the body of the vertebra, and then it can, kind of be set up like this. I'm not an artist, sorry guys. But then you have the spinal cord, and in fact, I drew this upside down because I don't have space, but hopefully you see what I mean. Let's erase all of this. So we have gray matter in the center of the spinal cord, white matter, which again are all axons, and then here's the central canal through which cerebral spinal fluid circulates. Each one of these little dots here is the nucleus of an ependymal cell. So the ependymal cells line the central canal. Let's erase this. And the ependymal cells, remember, have little tiny projections. And they act like cilia, and they help to allow the circulation of cerebral spinal fluid through the central canal up through the lateral ventricles of the brain. So the central canal is continuous with the lateral ventricles. Let's look at other glial subtypes. Here's a microglial cell. Remember, these are the guys that crawl around and clean things up. Here is an astrocyte. Look at those big astrocytes. Now, what I love about astrocytes is they can do one of two things. They can touch an unmyelinated piece of an axon. Here's an oligodendrocyte forming two pieces of myelin on a single axon. And so this is supporting the axon. Oops, let's get rid of that. On the other hand, here is a blood vessel. This blood vessel has lots and lots of end feet from astrocytes, basically plugging the holes right here between those endothelial cells. This is the blood-brain barrier. What that means is that things circulating around in the bloodstream cannot escape through these little gaps. And the gaps are just spots between two adjacent endothelial cells inside of the blood vessel. Here are the ependymal cells. Here are some of the long processes on one end, and they can have little cilia there sometimes. And so this is a picture from your book. This picture here is a picture I found somewhere else. And so this picture from your book also has the same type of organization and elements that will allow you to understand the function of ependymal cells, astrocytes, microglia, and oligodendrocytes. We can draw a little bit of these guys here. How come they don't have them there? Well, you don't always find them everywhere in the central nervous system. So we did talk about the Schwann cells a little bit. Schwann cells can also be called neurolemmacytes, although most people just call them Schwann cells. These are the cells that make individual pieces of myelin along the length of an axon. They surround the peripheral axons, and without them, without a Schwann cell or without myelination, peripheral neurons, the speed of electrical conduction down the neuron slows way down. You're going to hear about that big time in physiology. Satellite cells, or amphicytes, again, like I said, regulate neurotransmitter concentrations. They also regulate waste in the system, and they help support peripheral neurons in the peripheral nervous system. So here's an example of Schwann cells myelinating an axon. This is a peripheral ganglionic neuron. We'll look a little bit more peripheral ganglia in a later slide. And then here are the satellite cells kind of buffering them, protecting them. And then here is a picture showing you the process of myelination in a peripheral axon. So what happens is, as an axon grows in, a Schwann cell, a young Schwann cell, starts wrapping around, and it starts wrapping, in this case, in a counterclockwise fashion, forming a tight junction. It's not a tight junction, but a tight association between the Schwann cell and the peripheral axon here. This little spot that is unmyelinated, so let's assume that you have another Schwann cell upstream of this region here is called the node of Ranvier, and that is a super important part of an axon. The node of Ranvier is packed with what we call voltage-gated sodium and potassium channels. This node of Ranvier is what makes all the difference in conducting electrical activity down an axon. You'll hear more about that in physiology. Let's talk briefly about the astrocytes and oligodendrocytes in the central nervous system. Remember, the myelin sheath in the peripheral nervous system is made by the Schwann cells. All right, that's peripheral. So oligodendrocytes, let's start with these guys, since we were talking about myelin. Wrap around axons, creating the myelin sheath. But remember, the oligodendrocytes can myelinate more than one axon at a time. So it turns out that they create additional nodes of Ranvier in the brain. But remember, the oligodendrocytes are also helping to create a structure and a support scaffolding for neurons in the brain and the white matter in the brain as well. Astrocytes are the absolute most common types of glia in the brain. They do a lot of jobs. They contact neurons, they contact capillaries, they form the blood-brain barrier, they help give a scaffolding to the brain. You will find them in gray matter, but somewhat in white matter as well. They can, to some degree, repair damaged neural tissue, but what the astrocytes do is, if there is brain damage, the astrocytes start proliferating, they divide, they make more of themselves. They clean up the area, but then they sit there. They don't go away. And that's called an astrocytic scar. And so if there's a stroke and the neural tissue dies, well, that needs to be cleaned up. You can't have all that dead debris in there, right? That's nasty. That's actually detrimental to tissues to have this dead decaying stuff in there. Astrocytes come in and clean that up. They divide, they make more of themselves, right? But then they sit there. So even if there were stem cells that could generate new brain cells, they have nowhere to go because the astrocytes are already taking up the space. So once we lose neural tissue, brain tissue, neurons, the astrocytic proliferation does clean it up, but it inhibits the production of more new neurons. There's a lot of research being done on neuroregeneration these days, but that's one of the barriers that we know about, astrocytic scarring in the brain. So let's talk about neurons now. We've talked a lot about astrocytes. There are two cell types, again, in the nervous system, glia and neurons. We've already talked about the glia. Neurons are way bigger than glia. They don't undergo cell division. So once a neuron dies, it's done. You can't replace it with a neuron sitting next door. That'll just go through mitosis. And what's its main function? Communication. Neurons communicate with one another. They're the chitty-chatty kitties of the body. So I have a few pictures of neuron structure, basic neuroanatomy at the cellular level. The cell body can also be called the soma, okay? The cell body is the large region in the center. It contains the nucleus and basically many of the organelles, not all of them, but many of the organelles that you normally find in a cell. Dendrites are where information comes into the neuron. Now, you can have activation of the actual cell body, but dendrites are basically where information from another neuron comes in. All right? Dendritic spines are these little knobby ends or these little branches here. The more spines you have on your dendrite, the more talking the cell is doing to other cells. In other words, dendritic density reflects computing power in the brain. The axon is this long extension from the cell body to the synaptic terminals. The axon can be very, very short, a couple micrometers, or very, very long. Think about the nerves that have to extend all the way down to your big toe from the sacral regions of your spinal cord, right? That's a long nerve. So each nerve contains long, long axons extending that whole distance. So the axon is one long process from the cell body extending to the axon terminals. The axon terminals or synaptic terminals here are what are going to react to or interact with the next cell body or dendrites. So let's draw a few dendrites here. Make another cell body. See, remember me, not the artist? So this is info out into the next cell. There are neurotransmitters that live in the synaptic end bulbs, which are each one of these little bulbs here. And those neurotransmitters are waiting in vesicles to be released upon the activation of an action potential. And actually, the action potential looks more like that. I'm not drawing a heartbeat, Valerie. You have to draw an action potential. Now, other terms. Perikaryon is the cytoplasm around the nucleus. Perikaryon is going to contain lots of mitochondria and something else called Nissl bodies. Nissl bodies are lots of ribosomes. Some are fixed, associated with rough endoplasmic reticulum. Some are free and floating around. Something you do not find in a neuron is a centrosome. Why don't you find a centrosome? You find centrosomes in astrocytes. I'm going to let you think about that. And here's another picture from your book identifying each of the elements of a neuron. The only thing we didn't have in the last picture were the oligodendrocytes in the central nervous system. As you can see here, this single oligodendrocyte is forming two pieces of myelin along the length of the myelin sheath. And between those pieces of myelin are the nodes of Ranvier. So these two pieces might be associated with another oligodendrocyte. And then here's a synapse here. Although they did a better job drawing than I did. They have like artists that can work with him. Ooh, oh, this is so exciting, actually. Ah, finally, somebody's drawn this out. Here is the presynaptic neuron and axon. So we call this presynaptic right here. When you get to the dendrites, this is postsynaptic. I have never seen a book that's drawn that out. I've always had to do my bad art. See this? Well, I erased it because it was so, oh, I put it there. See, it's so ugly. That's so much better. All right, so let's talk about axons. Axons are, they start at the base of the neuron, okay? Axons can emerge close to the nucleus, although usually the nucleus is isolated in the center of the cell. The beginning of the axon is called the axon hillock. Why is that discriminated differently? The axon hillock is where the action potential, AP, starts. Just as a heads up, there's a different density of voltage-gated sodium and potassium channels at the axon hillock. Therefore, once the cell is activated and needs to send an electrical impulse, boom, the axon hillock starts that electrical impulse very, very quickly and very, very strong going down the axon. But you'll hear more about that in physiology. The axolemma is the cell membrane of the axon. Remember, lemma basically means like membrane. Axoplasm is the cytoplasm of the axon. That's easy. In the axon, there's a lot of mitochondria. There are a lot of enzymes and neurotubules in there. The neurotubules are a scaffolding supporting that long extension of axolemma. There are end branches or terminals. Telodendria. Go away. That is a really old term, and I'm not going to ask you about that. Synaptic end bulbs happen at the ends of the terminals. And then the synapse is the space between the synaptic end bulb. So let's blow up a synaptic end bulb here. Okay, dot, 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 dot. That's the axon here. Lots of vesicles containing neurotransmitter. Here's the dendrite of the postsynaptic cell. For those of you who had me in physiology, you know why I'm drawing it this way. And this space right here is this, is the synapse. Go there, so you can look at that again. So here is a picture, I believe this one's from your book, that is integrating everything except for oligodendrocytes. So let's put an oligodendrocyte here with some myelin. Put another oligodendrocyte here, forming more pieces of myelin. There we go. Okay, so you'd have numerous oligodendrocytes myelinating this long axon. There are some unmyelinated axons in the nervous system. Don't worry about that right now. And here are the synaptic terminals, and you can see this little space here, which is the synapse between the pre and the postsynaptic cell. They did this really well, actually, in this book. I'm impressed. The synapse can happen on the soma of the next cell down the line or on the dendrites. Now, there are other shapes of neurons throughout the body. The neuron that we typically talk about and that we were just talking about just now are the multipolar neurons, right? You have info in going down to info out, and they've stripped the myelin from each of these pictures so it doesn't look quite as confusing. You're only dealing with the neuron. You have the pseudo-unipolar neuron. Basically, what you have is two ends, but the cell body is in the middle and set off to the side. You find this very often in peripheral nervous ganglia. Over here is a bipolar neuron. It looks a lot like the pseudo-unipolar, but the cell body is exactly in line with the dendritic axon and the synaptic axon. So this is the dendritic axon. Here and here. And this is the synaptic axon. Right there, there, and right there. Now, one of the weird types of cells are the anaxonic neurons, where you don't see a clear axon. These cells may be actually initiating certain processes in a tissue. You'll find some of these in like the retina, in the heart, a few places. Most of the cells we're going to be talking about, though, in anatomy are going to be the bipolar, pseudounipolar, and multipolar. But you do need to know that the anaxonic neurons exist. And so in your book, they are just introducing unipolar, bipolar, or pseudounipolar, and multipolar neurons. Now, other classifications of neurons have their own special names. There are the pyramidal cells in the cerebral cortex because they look like pyramids. You have Purkinje cells. Look at this beast right here. Okay, that is the Purkinje cells in the cerebral cortex. That's a dendritic field. Look at that. And it has what we call axon collaterals. The axons branch off and go to other places. In the olfactory system, here's the olfactory epithelium, okay? You have olfactory cells extending axons down through the cribriform plate, but then they synapse onto other cells going up to the brain. Don't worry so much right now about the neuroanatomy about that. The example here is just that you have a functional group of neurons that are kind of organized in a way, how do I really want to put this? Basically, you have a bunch of cells that are specialized to do a specific job. And in this case, these cells, these bipolar neurons, are sending information in here, the dendrites, and sending it down to synapse on a ganglia here. Actually, it's up. Now, this is just, if we come back here, structural classification. Functional classification, different. We have afferent neurons in the peripheral nervous system. These guys are usually the pseudounipolar. This is just what we were looking at right here. That is an afferent situation. Stuff is coming in. Pseudounipolar or bipolar in this case, but pseudounipolar or bipolar, you're always going to find as afferent neurons. Efferent neurons are typically multipolar neurons. I'm trying to spit these words out. They are efferent. So afferent, pseudounipolar, bipolar. Efferent, usually multipolar. Interneurons are a total pain in the neck. Some of these are actually the axonal neurons. They are very different from afferent and efferent. They integrate information. They do what we call gating information. They accept information in, and then they tell cell 1 or 2 what's going on based on the degree to which they are stimulated. That's all you need to know about that. These three terms are the most frequently used terms in anatomy: afferent, efferent, and interneurons. However, you will be talking a bit about bipolar, pseudounipolar, and multipolar neurons. So you have shape and function. Oops. Now the neural impulse is the electrical conduction that jumps from one node of Ranvier to the next in a myelinated axon. Neurotransmitter release is the consequence of these action potentials or nerve impulse. A nerve impulse is the same thing as an action potential. You guys, I wish we could talk about it in anatomy, but it would take too long. Neurotransmitters are released once the action potential gets down to the synaptic end bulb. And then the neurotransmitter itself, the molecules that are exocytosed from the synaptic end bulb, interact with the dendrites, or in the case of voluntary muscle, the muscle cells themselves, to start the next process. So that's the end of this first part of the nervous system. We will be moving on to neuroanatomy in the next chapter.